Hello, I'm Bethanie Edwards, and I'm an assistant professor at UC Berkeley in the Earth and Planetary Sciences Department. My expertise is in lipidomics, and today I'm going to be walking you through some examples of how metabolites, more specifically lipids, have been used in conjunction with DNA and RNA to better understand complex microbial ecosystems and ocean biogeochemistry. This will be our outline. So we'll start with the central dogma of molecular biology, move on to the biological carbon pump and the microbial ecosystems that are involved. Then I'll give you a brief introduction into the different ohms, and we will walk through how you would extract and analyze a lipidome. And then we'll switch gears and talk a little bit about some recent studies that have linked DNA and RNA to lipids through integrative omics and said something about ocean biogeochemistry. So the central dogma of molecular biology dictates that DNA gets transcribed to RNA, which then gets translated into proteins. And these proteins include enzymes that do metabolic work throughout the cell. This is the complex microbial ecosystem that we seek to understand. Quantifying biomolecules is particularly powerful because these are largely single-celled microbial communities that are living and dying by the molecule. I will quickly walk you through the biological carbon pump. So we have phytoplankton, which are vastly diverse, photosynthesizing in the surface ocean, taking up CO2 and turning that into biomass. That biomass can be transferred up the trophic food web when phytoplankton are consumed by microzooplankton, like dinoflagellates and ciliates, or by mesozooplankton, like copepods. Phytoplankton can also aggregate together and sink out of the surface ocean. These sinking particles are colonized by bacteria and archaea on their way down and get remineralized back to CO2. These sinking particles can also be disaggregated by zooplankton grazers who continue to contribute to carbon export through fecal pellet production, diel vertical migrations, and ontogenic migrations that are seasonal, like diapause. Phytoplankton and other microbes can also be infected by viruses. We tend to conceptualize the impact of viruses as the viral shunt. So viruses infect a cell, burst it open, and move carbon from this living suspended POC pool into the dissolved organic carbon pool, which is more readily accessible to bacteria and can be easily remineralized back to CO2. But we know that some phytoplankton, when they're infected by viruses, actually aggregate together and enhance carbon export, and we call this the viral shuttle. The net result of these processes is that 50 gigatons of carbon is fixed in the surface ocean each year, and somewhere between 12 and 5 gigatons of carbon is exported to depth. And there's a lot of variability in that number. And because these microbial communities are so complicated, there's a lot of uncertainty in how much carbon is stored in the ocean, how long it's stored there, and how do we integrate microbial ecosystem dynamics into biogeochemical models. Next generation sequencing, advances in mass spectrometry, and the ushering in of the big data era have made it possible for us to look at the entire pool of DNA, RNA, protein, and metabolites from a sample and compare that to metadata such as geochemical measurements or classic microbiological techniques to kind of get a handle on this really complex microbial ecosystem that's having a big impact on how carbon is cycled through the ocean. And so through these omics, we're effectively following the central dogma of molecular biology by sampling across these ohms. So if we were to extract and sequence all of the DNA from an environmental sample, that would be the metagenome. And that could tell us who is there and what is their metabolic capacity based on what genes are in their genomes. If we were to extract and sequence all of the RNA within a sample, that would be the metatranscriptome. And if we focused in on the ribosomes, The ribosomal RNA, we could tell who's there and how active they are. We could also focus in on the protein coding regions, the mRNA that's being transcribed from the DNA, and that would tell us which genes are being expressed. And that could also give us some information about which taxa are doing that gene expression. Most integrative omics studies integrate between the metagenome and the metatranscriptome. That's fairly common these days. So mapping metatranscriptomic reads back to an assembled metagenome. We could also quantify and break apart proteins to figure out what their sequence of amino acids is. And that would allow us to say who's there, and also what molecular machinery, what proteins do they have in place. These 3 ohms are great for looking at community composition and general microbial processes, but we're not actually measuring metabolic rates or reaction rates by looking at these. To do that, we need to go one step down and look at the metabolome. So the metabolome would include all the small molecules that these enzymes are acting on, the substrates and products. And the lipidome is a subohm of the metabolome. And the reason why they're oftentimes considered separately is because metabolites are water soluble. They're floating around in the cytosol of the cell, whereas lipids tend to be hydrophobic and are either making up the membranes or they're sequestered away in specialized compartments within the cell. And so when we do the extractions, we actually separate the 2 of them. So if we wanted to sample the lipidome, we would do a blind Dyer extraction, which dissolves and breaks apart a cell by sonicating them in a mix of water and organic solvents. And then you add in more organic solvent later, and it causes a phase separation. And all of the lipids sink into or are dissolved in the organic phase, and all of the metabolites remain in the water in the aqueous phase. And the proteins are situated in this interface between the 2 of them. So a blind Dyer is how we would extract a particulate lipidome. And so these are lipids that are captured onto a 0.2 micron Durapore filter. Sometimes people use GFF filters. We could take the filtrate from filtering and put it onto a solid phase extraction cartridge and get at the dissolved lipids. And so once you have your lipid extract, whether dissolved or particulate, you have to analyze it. We use liquid chromatography and mass spectrometry to do so. So ultra high performance liquid chromatography would separate out the lipids based on how hydrophobic they are. We use reverse phase chromatography to do this. So the water soluble, more metabolite-like lipids would come off the column first, followed by more and more hydrophobic compounds. And then as these compounds are eluting off of the column, we measure their mass to charge ratio using a mass spectrometer. We can also apply a bit of energy and fragment each compound and measure the fragments that it makes. And this is how you do annotation, or more competent annotation, is by comparing the fragmentation of your compounds to spectral libraries that have spectra for fragmentation spectra for authentic standards, or in silico fragmentations that are predicted by models. And that would allow you to annotate a compound more confidently. And so this particular compound, we're able to say that it's a triglyceride with 50 carbons and 8 double bonds. And the 3 fatty acids that it has are a C14O, a C16-3, and a C25 fatty acid. Lipid annotation is not trivial. This is the hardest part of lipidomics and metabolomics. And this is because you're looking at thousands of unique compounds that you don't have an authentic standard for every single one, and they don't even make them. So a lot of times you really are relying on predicted fragmentations from in silico libraries. To give you a sense of the data reduction, so we might start off with something like 66,000 different peaks, different compounds that we've identified, we can subtract out the naturally occurring isotopes, and you're left with something like 46,000 different peaks. And then you can apply or search against some of these databases, and you might be left with something like 9,000 annotations that then you can go through and compare to a spectral library and get really confident annotations. So once you have your confidently annotated lipidome, you would move on to statistical analysis, usually using multivariate analysis or machine learning to tease apart these really large data sets. I'd like to give you a tour through the lipidome of Phaeodactylum tricornutum, which is a model diatom species, just to give you a sense for what types of compounds and what their biogeochemical importance is. So here, alluding in the middle of our chromatogram, are the structural lipids. These are the intact polar lipids. You can see here a hydrophilic head and then a hydrophobic fatty acid tail, 2 of them. And so these are the lipids that make up your membranes, right? So every cell on this planet has a membrane that's made up of a lipid of some type or another. And these membranes can be modified to deal with temperature stress, nutrient stress, pressure. They can also be chemotaxonomic and tell you something about broad taxonomic clades and their abundance in your sample. Lipids can also be used for energy acquisition. So things like pigments and carotenoids can absorb photons from the sun. And compounds like quinones can facilitate electron transport across membranes in both autotrophic and heterotrophic metabolisms. Lipids are mostly associated with energy storage. So here you can see hydrophobic triacylglycerides alluding kind of later in our chromatogram. And these are the types of compounds that diatoms store in lipid droplets and copepods store in lipid bodies. Lipids are also important for cell signaling, and I spent most of my academic career studying oxylipins, which are oxidized lipids, and their production by diatoms and how that impacts microbial ecosystems. We found that at low concentrations, these compounds can stimulate particle-associated bacteria and enhance remineralization. We also found that these compounds can deter grazing by microzooplankton on diatoms. And since the early 2000s, it's been known that these signaling molecules decrease the reproductive success of copepods that are feeding on diatoms producing these oxylipins. So lipidomics gives us a really wide analytical window to look at a lot of different compounds that can tell us various different types of information about the system. So it's a really powerful tool. A good example of where integrative omics across metabolites and lipids and transcriptomes has been done would be the diel studies that have been done in recent years at Station Aloha. So these experiments would be about 4 days long, sampling every 4 hours, which is exhausting. And I've heard firsthand from many people, just how strenuous this sampling scheme is. But the data that's coming out of it is really fantastic. I wish I had more time to talk about it. But there are 3 papers that are out so far looking at lipidomes or metabolomes and comparing them to transcriptomes, and also a meta-analysis that's taken all of this data and tried to kind of synthesize it down. And so the big takeaways are that most lipid and metabolite concentrations are peaking right at dusk. But there are a subset of molecules that are peaking in the morning, in the afternoon, and in the middle of the night. There tends to be an offset between gene expression and metabolite concentration. So oftentimes they would find that the metabolite would peak at dusk, but the gene expression, the gene responsible for the production of that metabolite, would peak in the mid-afternoon. So that really suggests that we really need proteomics when we continue these studies. Some of the biogeochemically relevant information to come out of this. This study by Kevin Becker in 2018 found that about up to 12% of primary productivity was being respired as tags at night. And using transcriptomics, they were able to link this to dinoflagellates and haptophytes. This paper by Kevin Becker in 2021 looked at pigment turnover, and they found a drawdown in photosynthetic pigments over the day, but not a decrease in primary productivity. So there's some implications for how we extract primary productivity out of satellite data. And this was largely a photo adaptation response of the community. And then Angie Boyson had this paper that just came out last month looking at metabolites and transcripts in the same body of water over the same time course. And she found that it appears as though Crocosphere and other prokaryotes may be cycling osmolytes as energy storage molecules. So this is something we really weren't aware of before. It's very interesting, exciting science coming out of these studies where you're integrating from the metabolome and the lipidome through the transcriptome. My lab was fortunate enough to get to tag along on a diel study in the California coastal ecosystem. A big thanks to Francisco Chavez and his group and the Monterey Bay Time Series for allowing us to tag along. We sampled the particulate lipidome through the water column at noon and at night over 5 days in early June in 2019. We found that the particulate lipidome as a whole did change significantly over the day. So here we're looking at a partial least square differential analysis, which compresses all the variability in our data into a few components and plots our samples against those components. Component 1 is actually separating our samples by depth, with the 20-meter samples clustering over here and the 0-meter samples clustering over here. But components 1 and 2 are separating our data by day and night. So the particulate lipidomes are quite different day and night. And these two, this day sample is from day 4. And we think that it's clustering with the night samples because there appeared to be a freshening event. So there's some rain on day 3. We think that that's the reason why it's behaving oddly. We ran a supervised random forest to look specifically just at day-night differences. We asked the random forest to classify all of our samples as either day or night, and it did an excellent job. It only misclassified one sample as night when it was actually a day sample. And we can look at the compounds that are significant for guiding the walk of this random forest. And I mean, this is all very preliminary, but some interesting things is we see this betaine lipid, which is associated with chloroplasts and known to, betaine lipids are known to vary dielly at station aloha. So we are seeing that being significantly more abundant in the day than at night. And we also see this wax ester, which has the opposite pattern. It has higher concentrations in the surface ocean at night than in the day. And wax esters are storage lipids used by heterotrophs. So we think that this might be a biomarker for diel vertical migrators. We're excited to integrate our lipidomic data with the DNA and the RNA data that hopefully will come out in the near future. But just a preliminary look at a diel study in a different ecosystem where there's a lot more forcing than the open ocean. In late June and early July, we sampled along the coast of California starting in Monterey Bay and making our way to Point Reyes, we found that Point Reyes was more silica limited, and the diatom community there had recently undergone a viral lysis event. And we found that Monterey Bay, the community there was less silica stressed, and the diatom community was actively undergoing viral infection, but they hadn't quite lysed yet. I also sampled the dissolved lipidome and the particulate lipidome across this cruise track. So we can ask whether the lipidome corroborates this story, and can lipidomics tell us anything new about the system? Here's a look at the particulate lipidome. And it kind of does what we would expect it to do. So here we're looking at the relative abundance of these lipid classes across our samples, so station and depth. And we see that the shallow samples are much richer in lipids, which is what you would expect with biomass being concentrated in the surface ocean. And we do see that the particulate lipidome at Point Reyes is significantly different from the particulate lipidome at Monterey Bay. So the richest dissolved lipidomes were observed at Point Reyes, and this is what we might expect from having diatoms being lysed open and spilling out their cellular content into the dissolved organic carbon pool. This is all very nice descriptive analysis of the data, but can we integrate across the lipidomes and also incorporate the transcriptomes that were collected along that cruise track in a more quantitative way, right? So we did this using this package called Diablo, which stands for Data Integration Analysis for Biomarker Discovery Using a Latent Component Method for Omics Studies. What an acronym, but it's a really great way of integrating across OMS. And so what Diablo does is it maximizes the common or the correlated information between multiple data sets and identifies the key omic variables that explain and reliably classify our samples as either Monterey Bay, being from Point Reyes, or being from offshore sites. So we can take all of these OMs, incorporate them into our analysis. Here we're looking at multivariate analysis of each omic data set presented by itself, but the actual PLSDA is actually informed by all of the omic data sets together. You can see that the dissolved lipidome is the most discriminant, with the 95% confidence intervals for Point Reyes and for Monterey Bay not overlapping. We can ask which feature of each OM was most significant for the classification of the samples. And here we're looking at which variables from each OM contributed significantly to the separation on component 1. And so we can see that Point Reyes is really distinguished from Monterey Bay based on, one, the diversity of the diatoms present in the 18S sequences. So you can see that Monterey Bay is really more Pseudonychia dominated, whereas all of these other taxa, specifically Navicula and Thalassiosira, were distinctive for Point Reyes. We can also see that reads mapping to these unclassified single-stranded RNA viruses were most associated with Point Reyes as well. And these are things that you could kind of like glean just from looking at the figures, right? But when it comes to the dissolved lipidome and the particulate lipidome, where there's a lot more variables, this type of analysis is really helpful. So we can see that in the dissolved lipidome, these oxylipins, these C20 and C22 oxylipins are characteristic for Point Reyes. And then within the particulate lipidome is these oxidized tags that are most associated with Point Reyes. To look at which of the variables across omic data sets were most correlated with one another, we used a Pearson correlation matrix. And so just to kind of orient you to the graph, we have the significant variables across the ohms organized here with a different color for each of the various ohms. And then lines between these variables indicate positive correlations with a R value greater than 0.95. And what we see from this is that the Navicula and Thalassia Syra 18S reads were significantly correlated with oxidized tags in the particulate lipidome, so the storage lipids, and potentially allelopathic oxylipins, so oxylipins known to have detrimental effects on members of the ecosystem, as well as some unclassified single-stranded RNA viruses. And correlation, is not causation. So this does open up a lot of new questions for us. We can't say for certain that it's the infection of Navicula and Thalassia Syra by this unclassified single-stranded RNA virus that's leading to increased oxidation of these tags and release of these oxylipins into the dissolved lipidome. However, we do know that these same oxylipins have been observed in culture experiments where we've infected Cattosaurus socialis with single-stranded RNA viruses. And I went into the dissolved lipidome and looked at the fragmentation of these lipids and compared them to authentic standards as well as to fragmentation done in silico, so from models. And I was able to annotate these oxylipins a bit further to get better IDs for them, and then looked in the literature and found that these oxylipins are associated with a decrease in copepod reproductive success. And this compound here in yellow, it's an analog to a compound that we know decreases microzooplankton grazing. So going back to the question of whether the particulate in the dissolved lipidome corroborate the original analysis of viral infection being active at Monterey Bay in a recent lysis event in Point Reyes, I would say the overall descriptive analysis certainly does. And then when we do this integrative analysis, we do find out something new about the system, specifically that these dissolved lipids are distinctive for Point Reyes, and they point towards some grazing deterrence going on in the ecosystem. We also have, new hypotheses about viruses that might be infecting Navicula and Thalassia Syra. And then these oxidized tags, we see these a lot of times in stress responses in culture. So these might be interesting to track down. And we typically think of dissolved lipid oxylipins. We typically think of oxylipins as being produced from membrane lipids. But the links between these suggest that maybe there's other pathways for biosynthesis of oxylipins from tags. I'd like to leave you with what I think are some of the major challenges and opportunities in the integrated omics space. And I'd like to thank my collaborators and in funding agencies and in graduate students who've helped get this data to the point that it's at today. And I look forward to talking with all of you in the panel discussion next week.